For my thesis, I researched RT-qPCR quantification of SARS-CoV-2 in wastewater, doing an in-depth investigation of standards. I did this under Dr. Robert Della Tolla from the Department of Civil Engineering and Dr. Richard Goulet from the Department of Earth and Environmental Sciences. With unpredictable case increases and new variants, there's a growing need for more effective tracking of positive COVID-19 cases. So currently there are different ways to test for SARS-CoV-2. The first is clinical testing. However, it's not well suited for mass surveillance because it can be costly, time-consuming, and vulnerable to resource shortages, so not every positive case can be detected. After discovering that the virus is fecally shed, researchers started looking towards wastewater-based epidemiology to more effectively estimate the prevalence within the entire population. And so in both cases, the main method for quantification is through RT-qPCR and the use of a standard curve, which involves the serial dilution of a known sample with the resulting CT values that are plotted against the log of the concentration. So what is the problem exactly? So many labs are using different standards to construct their standard curves, which range in their template material and in the methods in which they were obtained or generated. And because of the inherent differences between standards, different curves are produced, leading to variable results or either over or underestimation of viral signal. And so the goal of the study is to test the performance of commercially available standards and help inform one's choice for the most appropriate standard for their essay. So to accomplish this goal, we first started with a literature review to determine which types of standards were most commonly used. And so we used Publish or Perish to generate 183 applicable publications. They were separated based on wastewater and non-wastewater studies. The identified standards were grouped into 7 categories. So there was a couple RNA categories, a few DNA, and an unspecified. For the experimental component, we tested 7 commercially available standards, in which they were all serially diluted in triplicates for each standard. And these samples were analyzed by RT-qPCR, standard curves were generated, and then they were then used to quantify viral signal. The first figure shows an overview of the entire study range, in which 21% of studies were wastewater related, while the remaining 79% or so were non-wastewater related. So the second bar graph then illustrates the distribution of the standard types within the 2 categories. So the main takeaway of these results are that synthetic RNA and plasmid DNA were the most commonly used standards, which is very interesting considering how different they are from one another in their structure, their stability, and their amplification during RT-qPCR. And so in non-wastewater studies, synthetic RNA made up over half of the standards, while plasmid made up less than 30%. And in wastewater studies, it was pretty even between the 2, but plasmid DNA was the leading standard. Also, in wastewater, G-block standards were considerably more used than in non-wastewater. Now, as for standards, all curves are plotted on the same graph. So the components of the curve that are important to consider are the slope that represent reaction efficiency, and the y-intercept that represent reaction sensitivity, or in other words, the amount of cycles required to detect the lowest concentration. Although some curves seem to overlap, they are different, and so the difference in slopes suggests a difference in reaction efficiencies between the standards. Ideally, you would also want a standard with a lower y-intercept, as you'd want it to be able to detect the smallest concentration possible. But what exactly do these differences mean? Well, the slope and the y-intercept are the 2 parameters that are used to quantify viral signal. And so differences in parameters here with the standard curve translate to a difference here in the number of viral copies quantified. And so we took a sample data set from Ottawa's daily SARS-CoV-2 wastewater signal and quantified copies of the gene using each standard. And so the bright pink line represents daily confirmed positive cases, and the red is the actual viral signal that our lab is reporting to Ottawa Public Health. And so as you can see, using different standards can lead to different amplitudes in viral signal and also different interpretations in the community. So first and foremost, it's important to note the structure of SARS-CoV-2 as single-stranded RNA. And to get the most accurate reading as possible, the standard must closely resemble the sample material. So one of the main issues is that there's variability in the reverse transcription step of RT-qPCR where RNA is converted to cDNA before amplification. So DNA does not account for the variability within this step because it does not require reverse transcription, but RNA, on the other hand, closely mimics the sample template and can correct for the variation of the RT step, which leads to a more accurate quantification. And for this reason, an RNA template is the preferred material for the quantification of SARS-CoV-2. And then here are some of the references that I used for my research, and I just wanted to thank you for listening.